Welcome back to the Deep Dive. Today we are attempting to map out a truly complex internal battleground. Systemic Lupus Erythematosus, or SLE.
Right, and it's long been nicknamed the Great Imitator by rheumatologists.
It's such an apt name, isn't it? Because the symptoms are just all over the place.
They're wildly variable. I mean, you see painful, swollen joints, this chronic, just overwhelming fatigue, fevers, and of course that classic malar or butterfly rash across the face.
Which only appears in, what, about a third to maybe two-thirds of patients?
Exactly. So it's easy to mistake it for rheumatoid arthritis or chronic fatigue syndrome or even some kind of infectious disease at first.
And the context here is just so critical. For anyone listening, SLE is devastating autoimmune disease. The immune system is attacking healthy tissue.
And it overwhelmingly strikes women. We're talking a 9-to-1 ratio compared to men, usually right in the prime of life, between 15 and 45 years old.
So our mission for this deep dive is to really go beyond just listing symptoms and get into the surprising, almost paradoxical biochemistry that drives this disease.
We're going to drill down into two core systems that completely flip their roles. First, the immune complement system, which is supposed to be our protector.
And second, the very engine of our cells, our metabolism.
We're basically looking at how the body's own defense mechanisms become its most damaging foes.
Okay, let's start with that first massive contradiction then. The paradox of complement. Most of us think of the complement cascade as, the body's SWAT team.
Yeah, designed to puncture and destroy invading pathogens.
But in lupus, the literature reveals this shocking twist that involves, it involves clearance.
What's so fascinating here is that while complement activation is clearly responsible for causing severe tissue damage during a flare,
right,
a profound deficiency in certain complement proteins is actually the strongest predictor for developing severe lupus in the first place.
Wait, that sounds completely backward.
Yeah.
How can having less of a destructive system link to a worse outcome?
It all comes down to basic housekeeping. Biological trash disposal. It's been found that a homozygous deficiency of the early classical pathway proteins, we're talking primarily C1q and C4, is extremely strongly associated with developing SLE.
How strong a link are we talking about? Give us a specific example.
Okay, look at C1q. Almost every single human being identified who completely lacks the C1q protein. They've gone on to develop severe lupus.
Every single one?
Almost everyone. Now, you contrast that with a deficiency in, say, C2, which is associated with lupus only about 10% of the time. The numbers really do speak for themselves.
So C1 key isn't acting as a bomb here. It's acting more like a highly efficient janitor.
That is its critical function. The classical complement pathway is supposed to promote the rapid clearance and processing of dead or dying cells, that's apoptosis, and also large immune complexes.
The garbage removal.
Exactly. And if that garbage removal is defective, all that debris, which contains nuclear material and other autoantigens, it just accumulates. And when those autoantigens are exposed to the immune system, the system sees this mounting trash pile and launches the attack.
The body attacks what it couldn't clean up quietly. Okay, that explains complement as the friend. So let's flip the coin. Let's talk about complement as the active foe when the disease is flaring.
During active disease, it's the complete opposite. We can easily detect rampant complement activation in patients' plasma and tissues.
Okay.
We see complement proteins deposited in inflamed tissues, notably in the skin. That's the basis for the lupus band test, and very critically in the kidneys. That leads to lupus nephritis or LN.
And if we were to look at those inflamed tissues under a microscope, what are we seeing?
You're seeing the executioner, the membrane attack complex, or MAC. That's the final structure of the cascade, and it's designed to punch holes in cell membranes. The presence of MA is often really prominent in the most damaged areas. So if complement is causing all this damage, why is the standard clinical test measuring C3 and C4 levels in the blood? Why is it often considered a poor predictor of how active the disease actually is?
That's one of the great head scratchers for clinicians. There are a few reasons for that weak correlation. First, C3 and C4 are what we call acute phase reactants.
Meaning the body just makes more of them during inflammation.
Right, so production goes way up, and that can totally mask the fact that massive amounts of complement are simultaneously being consumed or hypercatabolized elsewhere in the body.
So the body's manufacturing new supply so fast that the blood test looks normal even while a storm is raging in the tissues.
Precisely. And the second reason is localization. The activation can be highly localized, deep inside the kidney's filtering units, for example, and it's just undetectable in the peripheral blood. You're checking the national supply when the local drain is clogged. Okay, now here's where it gets really, really interesting for me. The anti-C1q autoantibody twist. Lupus involves autoantibodies, but some patients, about a third of them, develop autoantibodies against a complement protein itself, specifically against C1q.
Yes. The body is attacking the very protein that should be clearing the cellular mess that started the whole problem in the first place.
It's unbelievable.
And these anti-C1q antibodies are highly associated with the most severe illness, particularly those kidney complications we just mentioned, lupus nephritis.
So the hypothesis is that C1q, after binding to those dying cells to clean them up, it inadvertently becomes part of the autoantigenic complex that the immune system targets. The friend gets tagged as the enemy.
It's a devastating example of a protective mechanism being completely weaponized against the body.
That is a profound failure of the body's security system. But that's all happening outside the cells.
Yeah.
How do we get from there to what's happening inside the cells? How do we jump from C1q to mitochondria?
Well, that's where the emerging field of immunometabolism comes in. We know that immune cells in SLE are running hot. They have these heightened metabolic demands driving their activity.
And the central hub for all that energy is the mitochondria.
It is. But before we dive into the mitochondria themselves, we have to talk about lipids because they're a huge clinical concern in lupus.
That's right. One of the biggest drivers of long-term mortality in SLE patients is accelerated cardiovascular disease, CVD, due to rapid atherosclerosis. And this is tied directly to dyslipidemia.
Dyslipidemia is just the clinical term for a lipid disorder. So you have elevated levels of LDL, the bad cholesterol, triglycerides, total cholesterol, and at the same time reduced levels of HDL, the good cholesterol.
And the risk isn't small, is it?
No. The risk of ischemic heart disease for someone with lupus is about 10-fold higher than in the general population. It makes CVD the most common cause of death.
And to complicate things even more, many of our conventional treatments, especially high-dose or long-term corticosteroids, can actually make the dyslipidemia worse.
It creates a really difficult balancing act in clinical care. And this lipid disorder doesn't just affect the arteries, it affects the immune cells directly. Dyslipidemia changes the function of what are called lipid rafts within T and B cell membranes.
I remember lipid rafts being critical for cell signaling. They're like little communication hubs on the cell surface.
Exactly. So by changing their composition, the altered lipids actually strengthen T cell activity, making them easier to activate. And in B cells, this distortion leads to increased spontaneous proliferation and, you guessed it, excessive autoantibody production. The engine is just running way too hot.
Okay, so let's move to that central power hub, the mitochondria. We're now seeing mitochondrial dysfunction as a key element driving this entire inflammatory cascade.
The foundational problem is altered mitochondrial homeostasis. We see defective mitophagy, that's the process for clearing out damaged mitochondria, and we see impaired mitochondrial DNA.
And that breakdown is linked directly to the most reliable molecular hallmark of SLE, right? The type I interferon signature.
The IFNI signature, yes. It's an upregulation of type I interferon-regulated genes, and it correlates perfectly with disease activity.
This is where it all seems to collide. The clearance failure, the metabolism, the inflammation, it creates this vicious loop. Walk us through how that self-sustaining cycle works.
Okay, so it starts with oxidative stress inside the cell. When you have reactive oxygen species, ROS, combining with that defective clearance process, mitophagy, you end up with oxidized mitochondrial DNA, or OXMT-DNA.
And this damaged DNA gets out of the cell.
It escapes the cell and starts circulating. This circulating OXMT-DNA acts as a damage-associated molecular pattern. It's basically an urgent distress signal.
And the immune system picks up that signal.
Picks it up loud and clear. It activates key immune cells, specifically plasmacytoid dendritic cells, or PDCs, through their toll-like receptors. This activation then pumps out massive amounts of IFNI.
And the IFNI just pours gasoline on the fire.
It really does. IFNI triggers a process called netosis. This is where neutrophils essentially commit suicide, extruding this biological web that contains proteins, DNA, and critically, even more OXMT-DNA.
Wait, so the IFNI makes neutrophils release more OXMT-DNA, which causes the PDCs to produce more IFNI, which triggers more netosis. It's a perfect self-sustaining loop of pathology.
It's a key mechanism that explains why the inflammation is so incredibly difficult to switch off. And we see the consequences directly in places like the kidney in lupus nephritis.
Let's see mitochondrial problems right in the kidney cells.
Yes. Mitochondrial abnormalities, specifically excessive fission, are observed right in the renal podocytes, the filtering cells, and they correlate with fibrosis and scarring.
So given this deep understanding of the broken engine and the failure to clear out this oxidized junk, What are the most promising therapeutic implications? Are researchers trying to target this cycle directly?
Absolutely. The research is really centered on breaking this metabolic loop. They're looking at cell-free mitochondrial DNA, CFMT-DNA, as a potential non-invasive biomarker. And interestingly, antibodies against mtDNA have been found in a significant number of patients.
So what experimental therapies are showing promise in trying to fix that broken mitochondrial engine?
Several drugs are being looked at. Metformin, which inhibits a mitochondrial enzyme complex, showed promising results in a murine lupus model. It increased renal function.
And sirolimus?
Sirolimus is an mTOR inhibitor. It's known to restore mitophagy, and it's already being trialed for patients with persistent SLE activity.
The most recent data on a drug called UMI-77 sounds particularly intriguing, though.
It is. UMI-77 is a potent mitophagy inducer. In lupus-prone mice, it reduced glomerular inflammation, T-cell infiltration, and immune complex deposits. But here's the key finding.
Okay.
The effects are achieved by restoring mitochondrial homeostasis in the plasmacytoid dendritic cells, the PDCs, not by directly repairing the damaged kidney cells.
Wow. So you're saying just fixing the immune cell's engine was enough to halt the runaway inflammation that was damaging the kidney?
That's what the data suggests. It's incredibly exciting to see these metabolic targets emerging, but we have to reiterate that the cornerstone of treatment is still conventional therapy.
Right. NSAIDs, corticosteroids, powerful immunosuppressants like cyclophosphamide, and the absolutely essential hydroxychloroquine.
And we know hydroxychloroquine is critical, not just for inflammation, but because it has favorable effects on lipids, reducing that cardiovascular risk and improving overall survival.
So the major takeaway from this deep dive seems clear. SLE is not just some generic autoantibody attack. It's a pathology rooted in this profound interwoven failure of the body's essential cleanup mechanisms.
Both extracellular clearance, the complement C1q system, and intracellular clearance mitophagy are derailed, and that leads to the accumulation of dangerous debris.
Apoptotic cells on the outside, oxidized mitochondrial DNA on the inside, and that debris is what initiates and sustains that relentless inflammatory loop.
We saw that the earliest genetic failure, that C1q deficiency, strongly predicts lupus because cellular clearance fails from the start.
Okay, so here's a final thought for you to chew on. We know that powerful protective role of C1q in clearance, but we also know C1q deficiency is very rare. So what unseen common mechanisms might be failing in the majority of SLE patients who have normal complement levels? What else is struggling to clear those dangerous autoantigens, allowing them to escape disposal and kick off that whole metabolic-driven cycle of inflammation? What other internal trash collectors are breaking down? That's something to consider until our next deep dive.